Next up, I'm going to give you a quick introduction to BigQuery, and then we're going to use BigQuery and Jupyter Notebooks to analyze genomic data. So first, this quick PowerPoint, we're going to discuss a little bit about the BigQuery Cloud Console, as well as Terra's in-app functionality for BigQuery, and then we'll do some hands-on exercises. So we'll get you back into your cluster really quickly. So don't delete it. Don't pause it, just leave it. So first of all, what is BigQuery? How many of you out there have heard of or used BigQuery before? Show of hands. 1, 2, 3, 4. Really? Only 4 people have heard of BigQuery before? Okay, well then this will be pretty interesting for you guys. So Google Cloud put out a statement that BigQuery is a fast, highly scalable data warehouse product with built-in machine learning that is fully managed and run by the Google Cloud Platform. So what that means is it's the equivalent to Google Cloud Storage. So we have these storage buckets that contain all of our BAM, GVCF, VCF files. It's the equivalent to that, but for tabular data. So for any of you who may have worked with databases before, like SQL, it's a database that exists on the cloud that allows you to query that genomic data. So for example, the ISB CGC platform hosts the TCGA dataset in BigQuery. So you can access that data through BigQuery. In addition to that, I have this whole list of genomic data there on the left. So the 1,000 Genomes project, which is what we'll be working with in our example, is in BigQuery. There's the Illumina Platinum genomes, the reference genomes, a whole bunch more there. And then over on the right, we have some annotations. So these we distinguish from genomic data sets in that the annotations contain more questionnaire type data, phenotypic data, things that don't necessarily contain genomic information but are still important for health studies. So you've got a bunch of annotations there. And a little plug for BroadEs. If you have data that you want to move to the cloud, Bits can help you do that. It's a perk of working here. They want to move us to the cloud. Any questions about this so far? Do you see any data sets you're interested in? No? All right. So one nice thing about BigQuery is that it allows you to get fast insight and results. Databases can be historically slow, especially if you're working on like really sluggish old machine like I did in college when I was trying to query a database. But in the cloud, you can query all this range of genomic information of these very, very large research projects in seconds. It's very efficient, and it's a better way of searching databases than simply drawing small samples and searching those at a time. It's faster than non-cloud queries on the order of seconds versus tens of seconds. And when you couple it with a Jupyter Notebook, you can do real-time data analysis on data that still lives in the cloud. So you're not downloading it to your local machine. You're still working with it where it lives. As far as functionality, BigQuery has some integrated analysis tools. If you're familiar with SQL or R or Python command line tools, things like that, you can use those with BigQuery. We have this big list here on the right of some SQL functions. This is not a comprehensive list, but there's a link to some more SQL functions that are available to you. But you can use all of those in order to narrow down your query to access the right amount of data that you want to do. And then you can scale to any project size, so process genomes in parallel or as many experiments as you like in parallel, and perform these really complicated queries. And I want to emphasize that it's on petabytes of data. There's just so much genomic data we have now that we need this really efficient way to access it. Speaking of access, there are 2 ways that we'll talk about today where you can get at this data. One is through the BigQuery console, which we have a screenshot of here on the right. That link there in the slide will take you to the BigQuery console. It's a great system that allows you to dig into data sets. You can check your query syntax. You can estimate costs for your queries using that. But the one we're going to be demonstrating today is Terra's in-app functionality. So I would say that this one's a little bit more user-friendly if you're not an SQL expert. You can explore questionnaire type data. You can generate custom cohorts, which is what we'll be doing. And then take that data and export it into a Terra workspace. So you can do notebook work with it. You can do workflow work with it, things like that. And with that, I'm going to jump off the slides. For those of you looking at the PDF there are some further slides here, which are going to walk you through exactly what I'm going to walk you through in person. But if you ever need a reminder of what I did, feel free to check those out. There we go. So I'm going to have you go to the home page again, which is app.terra.bio. You should be presented with this screen. I'm going to have you go to this View Examples button. So again, we're going to the Showcase and Tutorials page in the library. And what we're going to do is clone another workspace. So we just went through a Jupyter 101, and now we're going to do a quick start here. So you should find it, sorry, I clicked on that too fast. In the featured workspace list, 4 down is this Terra Quick Start workspace. So I want you to go into that workspace, and then you'll click on the little 3 dots in the upper right corner and select Clone. So I'm clicking on the workspace, and then we have these 3 dots in the corner, and we're going to clone it. So I'm going to append it with my name so that I remember it. And what I want you to do is use the same billing project you just used for your previous notebook. So if that was the free credits, use that. If it was the workshop-temp, use that. I'm going to show you a neat thing with that. So we, for my part, used help-ghk. So I'm going to go ahead and click Clone Workspace. And it'll load for a second. Is anybody having issues cloning? Please raise your hand. One of the TAs can help you.
Yeah. While we're waiting, so is all the data that's in Terra in BigQuery? That's the format in which it's saved?
No, not necessarily. So talking a little bit about the data in Terra all exists in Google buckets. So that's the Google Cloud Storage stuff. These files are tabulated, I'd say, in BigQuery so that you can find and access the right types of data sets. Yeah, and then a third place where you can store data that we just talked about was your notebook runtime. So once you have your workspace cloned, it should look like this, where you now have access level of owner. Mine is project owner because I own the billing project, so I have a bit higher permissions, but this just indicates that you have the ability to do things in this workspace. So one of the things we're going to do is go over to the Notebooks page. So you just ran through an intro to Jupyter Notebooks, so I'm going to skip that for you. And we're going to go to this R environment setup. Now, you may get this pop-up that says this notebook has been edited recently. That just means it was freshly created, it was freshly cloned. So I'm going to go ahead and close that warning there. And if you clone to the same billing project that you used before, you'll notice that your notebook runtime is still running, and you're able to just start up the notebook pretty quickly. It takes, I think, maybe 30 seconds to copy over the notebook information so that you can open it, but it was much faster that time. So when we talk about this shared runtime environment, it's a virtual machine out there where previously we had opened up one notebook, now we closed it and we're opening up a new notebook, but the computer itself still exists, the virtual computer, so to speak. Yes?
I've done this with both times I've opened this notebook. I get this next to where it said a warning about this notebook had been edited recently. Underneath it says, this Google bucket is associated with workspace that's currently not available. This should be resolved shortly. Is that
Okay.
comment?
Are you on the free credits?
Yeah.
Yeah, I think it might be because your free credits are new and you just created the workspace. But if you have any issues with it, like it throws another error, do you know more?
I've seen that error sometimes, and like it's usually like a lag thing, and it'll go away. When you make a workspace that's brand new, sometimes it says like you can't access the bucket for a few minutes, and so you might just be seeing that.
I see.
And then as far as the error that says this has been edited recently, I know it's weird considering you've never opened it. So I think that was one of the things we have open, working on rewording that so it doesn't seem like someone was working in your notebook when they weren't.
Got it. All right.
Okay, great. So has everybody opened up this R environment setup notebook? Okay, so I'm going to use one of the buttons that Anton just taught you about. Go up to the cell menu, and I want you to hit Run All. And what that does is it's going to execute every single cell in this notebook. And you'll notice on the left in the table of contents, it highlights everything in red. Those are cells that have been, we've told to run, but they haven't finished running yet. And gradually the red will fade away as they complete. So now the table of contents is completely cleared, and that means my entire notebook is done running. So what did we just run? Well, the reason why I clicked Run All on everything is because this is a setup notebook. It's designed to install the right packages we need for our work and basically set up this virtual machine environment for us. It's not terribly interesting, so I'm going to quickly skim through and show you a bit of some of the commands we ran. So the first one is to install these R packages. We have this command if missing, so that if it already exists, it doesn't try to overwrite it. Installs some tools from GitHub, these developer tools, and this folding extension, all these different things. These are some R packages that it checks to make sure exist in our workspace. And then one that came up recently was what's in my workspace. Down at the bottom, this devtools : session info. So again, devtools was a package we loaded up here at the top. But it tells you all of the versions and packages that exist in this particular virtual machine environment. I believe mine is a little bit more extensive simply because it's been, I've had this runtime environment for quite a while. So yours should list all the things you installed just now, as well as any other default things that exist. Any questions on that? Still loading. The notebook?
Everything's red.
Oh, still red. Okay. All right. I'm guessing mine ran quickly because it's already installed everything. Yep.
This is something you have to do every time. Is it linked to the account that you're?
So it's linked to the runtime environment. So we talked about pausing your machine would keep everything set up already. But if you deleted the runtime environment, then you would have to rerun this particular notebook.
What if you want to use a bioconductor method, which is an R? Do you need to do it in 2 steps? Or like, do you specify?
So the notebook we have here, is an R setup.
I don't know a lot about bioconductor, but I know that it's something that we're working to enable. It's in our quarterly goals.
Okay.
So I don't know much more than that, other than it's something that we're trying to enable. We use it.
All right, so for anybody who couldn't hear or the cameras at home, bioconductor isn't currently available, but we are working on it this quarter. All right. Any other questions? How's your script going? Is it still red? Running? One yellow? Okay, so the yellow indicates that that's the cell you have selected, so that's good. So we should be good to move on. So I'm going to go ahead and exit this particular notebook. You can do that in several ways. I'm just going to click the X in the corner, and that should take you back to your notebooks page. So there's an X in the upper right corner. Click on it, and it'll take you back to the notebooks page. You can also just click the notebook thing in the header. But we'll do it this way. Now, the notebook I'm going to run you through is this retrieve a cohort using BigQuery. So you can go ahead and click on that to open it up. And this is another case of, I'm going to walk you through everything so you can skip the screenshots. So I just had you open it up so we can come back to it easily. But what I want you to do is go back to the home page of Terra. I just opened a new tab so that I still have the notebook open. And this time we're going to go to this Browse Data button. So we've looked at two example workspaces, and now we're going to go to the Browse Data section. So that should take you to this datasets page in our library. Feel free to look around. There are tons of datasets here, all available for you to browse. The particular one we will be using today is this 1,000 genomes low coverage. And we're using it because it's a public data set, so you don't need any specific permissions to access it. Yeah?
So the BroadE data set here, so we have stored on the BroadE server our personal genomic information. We can transfer that over and use that in Terra.
So you would need the data to exist in a Google bucket somewhere. It can't access like an on-prem cluster, if that's what you're asking. Yeah.
Yeah, so like the server that's, I don't know, one of the main BroadE servers that we store all of our genomic data on.
Okay.
You just have to like basically copy that over into Google in order to use it in Terra.
Yeah, so in order to work in the cloud, the data has to be in the cloud. So you'd need to transfer it over. I believe Bits can help you with that. Yeah. Any other questions? Okay, so we're going to go ahead and hit this Browse Data button for 1,000 genomes low coverage, and it should take you to this page here. So this page shows you a whole bunch of annotations that exist on this particular data set, such as samples overview, gender, what population it comes from, the percentage that are 20x coverage, things like that. And what we're going to do is everybody is going to create their very own cohort. So right now we have 3,500 participants in this cohort, and I want you to make a cohort that's about 100-ish samples. And how you're going to do that is by simply selecting any of these bars in any of these charts.
Sorry, can you just tell me the name of where to get to this?
Yep, so if you go to the home page, it'll be in Library and then under Browse Data, under the 1,000 genomes low coverage one. And that takes you to this page. So to create a sub-cohort, which is what we'll be doing here, you simply click on a bar and it'll start narrowing down your participants. So I selected from the superpopulation East Asian, which consists of 617 participants. And it added the superpopulation equals East Asian to my query. So it's building a query for us using this neat interface. So that's still too many for me, so I'm gonna subset it to maybe 0.7 to 0.8 of the cohort is 20x coverage. And you can feel free to subset it however you like. I'm just gonna say that that number is good for me for now. Has everybody managed to make a cohort? Still working? Yeah? You can't see the data set? Did you click on low coverage? Because the high coverage isn't, got someone helping you.
Is there a good number to have in our cohort target just for what we're?
Around 100. Yeah, just so it runs pretty quickly. All right. Has everyone managed to select a cohort? Raise your hand if you've selected a cohort. Okay.
You select two.
Two things. So I just selected one from the superpopulation, and then I just scrolled to a different one and selected another. Or are you asking how to select two from this?
Within samples, within population, if I wanted two of those.
Just select both of them. Just click on both. So I just clicked on another, and it just added those samples. All right, so what we're going to do now is click this Save in Terra button. And we're going to name the cohort something so that we remember what it is. So I subset to the East Asian population with 20x coverage, so I'm just going to title it that. And then I'm going to hit Save. So name it anything you like, but something you'll remember. And then we're going to pick a destination workspace. So this will be the workspace that you just cloned. So it starts with Terra Quick Start Workspace. I have a bunch of those, so I'm just going to type my name because that's easier for me to filter on. And then you'll hit this Import button when you're ready. I like to leave it on this screen for a bit so people can see. But go ahead and hit Import. So once you've imported, it will take you to the Data tab of your workspace. And I believe this is the first time today we're encountering this tab, so I'm going to talk about it a little bit. It's created this new table for you called Cohort. And if you click on it, you can see your cohort ID is the one that's populated in there. So mine was East Asian 20X. You can see what data set it came from, as well as the specific query it would run in BigQuery. So if you're interested in what that particular text looks like, it's right there. And if for whatever reason you didn't name it in a way you'd remember, or you want to see what's in that data set again, just check this little box and click Open in Data Explorer. It'll open in a new tab and it'll show you that screen we were just on with all the bars you clicked on and all of the filters you have in the top bar there. Does that make sense?
Where was the Open in Data Explorer?
It's right here. So in the Data tab, I've clicked on the cohort table. And then it'll be grayed out at first, but all you need to do is check next to whichever cohort you want to do. We only have one cohort. Is my mic wrong? We're good? Okay. So we only have one cohort for now, so it's the only one in there to look at. Okay. Now that we have our cohort, we're going to go back to our notebook and actually work with it. So you should just be able to tab over to your 3_retrieve a cohort using BigQuery, or you can just open it again by going to the Notebooks tab in your workspace. Now, I just walked you through, all the screenshots in section 1. So I'm just going to click this little arrow and collapse it because we don't need to see those again. So I just clicked this little arrow here next to the thing to skip ahead. So I've walked you through the whole process of getting your query loaded into the workspace. The next bit is a little bit more of setup. So we ran that R setup notebook, which is good for the entire notebook, but there are some specific things that are needed additionally for this notebook. So I want to run these next 3 cells to get those installed. And all I'm going to do is select the first cell I have and click Shift Enter, Shift Enter, Shift Enter, Shift Enter. And it's going to run those 3 cells for me. And one thing you'll see pretty quickly is that I can run a bunch of cells in order. And while the second command is waiting for the first command to finish, it'll show a little asterisk next to it saying it knows it needs to run, but it hasn't run yet. And then when that command has finished executing, it gives you a number inside the little bracket. So I'm talking about this part right here that says ln1. It gives you a number inside those brackets to tell you which order it was run in. So in that way, it can be helpful to see what order you're running commands in, if you need to go back up and rerun it, things like that. So what these particular commands loaded for us, in particular, is a library called Reticulate, which is a library that allows us to use Python code inside an R notebook because we're just that complicated. We wanted to use some Python commands and we wanted to do some plotting, and R is a great language for plotting. So speaking of plotting, we loaded ggplot, and another library we loaded was for BigQuery, written in specifically the R language. So we'll be using those 3 libraries. Another module we loaded is called the FireCloud Service Selector, or you might have heard it called FIS. This was originally written for our previous platform called FireCloud. Now FireCloud is part of Terra. So it does essentially the same thing for Terra as it did for FireCloud, in that it can access some API calls, so Application Programming Interface, which I didn't know that was what that stood for. That's cool. So these API calls allow the applications to talk to each other. So the notebook application that we're in, this virtual machine for the notebook, and the workspace itself. And then the last bit is setting some variables so that we can use them. Speaking of variables, there are some additional project variables we're going to set up. So in section 2.2, click Shift-Enter 3 times to run those 3 cells. And what those 3 cells are doing is setting up these environment variables for us to use. All of you have cloned workspaces, and all these workspaces have their own IDs to them, their own names, their own unique identifiers in the terminal, things like that. So if we tried to hand you this notebook without giving these variables, you'd all have to change everything to specifically reference your workspace. But by using these variables, we've set it up so that I can hand you this notebook and you can run with it, or you can hand it to your colleague and they can run with it, things like that. So that's what this section is doing. And now we're finally ready to work with our cohort. The next bit is we're getting our cohort query. So we're creating these Python versions of our workspace variables because we're in R and we want to use some Python variables. So click Shift-Enter on this first command in section 3. And that should execute without printing out anything. So that's fine. The next bit is telling our FIS service selector to go into our workspace, grab that data table with the cohort in it, and create a variable for it within our notebook. So we have this thing in our workspace, and now we're bringing it into the notebook. So just hit Shift-Enter on that. There's a little note at the bottom of that cell that says, if you get an error running the cell, try rerunning. We've had an error in the past, and it was patched, so I don't think you should hit anything, but we left it in there just in case. It's a transient error, and you'll just be able to rerun it. All right, so let's see if all of that worked. So in the sanity check section, we're going to call the cohort entity variable we made by just hitting Shift-Enter, and it'll print out some information about what that variable references. So if you look at it, the first section is query, this whole section here, which is great because that is exactly what we saw in the data tab, or well, we only saw the first, maybe this bit of it, but if you had expanded it all, this is what the whole query looks like. It's interesting to me to see this query because this is the text that you would actually go to the BigQuery console and type in and execute, and that interface we interacted with created this query for you so that you know what you're getting and you're able to be confident that this particular command will get you the type of data you want. It also gives you the Data Explorer URL, so that's the UI interface if you want to access that. It tells you what the data set is and tells you the name of the cohort, which is interesting. This second line here, I'm going to hit Shift-Enter to run, is called Cohort Query. So all that was pulling out of this list just specifically this variable, so it makes sense that those two match. That's our sanity check, that they look the same. Does anybody have questions on this so far? All right, great. All right, next we are going to call BigQuery on our cohort. So what this does is it takes that command we have for BigQuery, it executes it and gives us back the information from that query. So hit Shift-Enter to run that one. And when I say it returns the results, it gives you a table in R, so it won't actually print the results as a result of that cell, but our next command will print the output, so you can look at the table. So when that's finished running, you can go ahead and hit Shift-Enter on that print line to see if it grabbed the right data you were expecting. Excuse me. So the print parentheses TT line prints out a table. It tells me that it's 266 lines long, which makes sense for me because my cohort was 266 participants. Yours should say something different depending on what particular cohort you selected, or if you selected the same thing as me, it should say the same thing. And then it begins to list some participant IDs. So it tells you all of the IDs of participants that are in that data set. Does this make sense? All right. So the next step is a query that's going to add gender data from the annotations to our list of participant IDs, so that we'll be able to tell that for sample HG00881 that was male or female. And the reason why is because we're going to graph it so we can see information about it. So if you hit Shift-Enter to run that query, it's saying for every distinct participant ID and gender, we're going to grab it from this 1,000 Genomes project, which is the one we are querying in the first place. And it's going to create a table for it and print out the dimensions of the table to check. So this one just grabs all the gender data from the 1,000 Genomes project. So it's not subset specifically to our cohort. So it's going to print out this 3,500 number saying that we have 3,500 unique participants inside this sample, or inside this data set. I shouldn't say data set, database. So that makes sense. And now we're going to, with the next line, merge the table with the gender data with the table that we have for just our cohort. And you should see that the dimensions of the merged table are just specifically your participants. So it's now added the gender data to your table, but it doesn't need to care about the other 3,300 for me-ish participants' gender data. Then the last bit here is to plot it. So it's going to look at gender versus, so male versus female for all of your participants and graph that number. I'm just going to zoom out real quick to show this whole table here. So for mine, it looks like it found 146 males in the cohort and 120 females in the cohort. So that's what the gender is there. So was everyone able to plot? How many of you have an exclusively male or female set? Yeah? Did you select male or female from the cohort? Makes sense then, that's good. So the big reason why we walk you through this BigQuery example is because very often data in the cloud can feel far away. Like you can't interact with it without downloading it, or you can't just quickly look at a file to see some information about it. But using BigQuery, you can pull the data into a notebook and query it, look at some examples, or you can perform some analysis with it. We've just shown plotting, but you can do all sorts of things with it. Whatever you need to do with your cohort, you can do in this method. Yeah, question?
So I had selected a key station population,
Mhm.
Japanese, and the gender distribution was some, I don't know, 40 females and whatever, some 50 males.
Sure.
And it was total 105, but it is all in the plot, it's showing only females.
Showing only females. Can we look at that?
If you scroll, maybe it's just too big. Just
All right, someone's gonna come look at that for you. So that's it for my BigQuery presentation. I wanted to show you that in your notebooks section, there's one other notebook in here called Read Data from BigQuery. It walks you through a similar example showing how to create a query outside of simply using that interface. So we have this SQL query that you can run. I'm not going to run you through it today simply because we don't have enough time to run through all of it, but all of the information in there is self-explanatory, so you can definitely run it on your own time if you are interested in seeing a bit more about how BigQuery works and how it can work with your data. So with that, I wanted to open up to questions. Any questions on anything we've done this whole morning related to notebooks? Yeah?
Kind of general question
Sure.
about the notebook and workbook.
Sure.
How can you share it with the others? And then also kind of, I think the workbook and workspace can be the cloned or copied and with others, but the regarding the kind of the notebook itself can
Sure.
be kind of the shared or copied.
Yeah.
And the other question is related to that. So is there any kind of version control like GitHub so that we can make a branch, especially when we share with others?
Sure. So the first question was, how do I share a notebook? There are many ways to share a notebook, ranging from you can simply download the file and give it to your collaborator. If you want them to work on it within your workspace, then you'll simply share that workspace with them. You can give them permissions to be able to open up your notebook and run work in it. As of right now, and somebody correct me if I'm wrong, we don't have version control in notebooks. But what I would recommend is if you want to take a timestamp of a notebook, copy it. There's a button for just copy this notebook within the workspace, make a slight change to the name, and then do that. It is on our goals to get that done, I believe, very, very soon. Yeah?
Generally speaking, users can go to the terminal and use their own version control using Git, correct?
I'm not sure. So the question was, can you go into the terminal and have your own version control using Git?
I don't think so, because we don't, it's not into a GitHub repo or anything like that. It's just a pure Google bucket. So there will be new feature rolled out, which is for making sure that you're not following say someone else is editing your notebook. You guys will see that this notebook is locked by someone else. You won't be able to, follow each other's changes.
Can you write custom queries which would include more than one cohort?
More than one cohort, yes. So if you go back to the data browser, Data Explorer, really quick, I can just show you that. So if I zoom in so you can see and go to the Dataset Explorer, I'm going to go to the 1,000 genomes. Now let's say I wanted to make a different cohort and import it into the same workspace. I'm just going to select two things randomly and then save the cohort in Terra. So I'm going to name this cohort 2. Hit save, zoom out a little bit, and then I'll import it to the same workspace. I'm doing this really quickly so I can just show you what it looks like when it's there. And what that does is it updates your cohort table, and now it says 2. So that means there are 2 things in here. So this second cohort here has a different query associated with it, a different workspace URL. But if I were to go into that notebook we were just in, and if I wanted to run this same analysis on a different cohort, all I have to do is go to sorry, I'm looking for like a parentheses 1 that we have in here somewhere. Aha, here. So so for me it's line 8, it's in section 3. There's this cohort entity and cohort query where the FireCloud Service Selector is going to BigQuery and getting and building the table for us. There's this line that says cohort entity bracket 1. If I change that to 2, it will do the second cohort in my table. And then you can run it on all the other stuff. Yep. I don't know. I have never tried. Does anyone know in the room? Yeah, you might be able to just create a new cohort with the join of them. All right, any further questions? All right, well, great questions, guys. We're going to dismiss for lunch, and we'll be back at 1 p.m. We're going to start up with a talk about billing and access, so talking about that sharing a little bit, as well as how cost comes into play and how you can manage it. And then this afternoon is going to mainly be focusing on workflows. So we've talked a lot about notebooks, and now we're going to talk to you about pipelines and how you can use them and how you can run them.